and the female, and they're different on the inside. When we look at nematodes, how we determine if they're there, we're commonly looking for their eggs or the ova. For example, ascarids, one specific type of nematode, has a very thick, round to oval type of egg. It has a very thick outer shell. That shell can be rough or it can be smooth. On the inside of the egg, you see a single cell stage for ascarids. So this is an ascarid egg. Okay? Other types of nematodes like strongyles, okay, or trichostrongyles, they have an oval or elliptical shape of an egg. Their shell is really thin and smooth, but when you look inside of the egg, it looks like a morula. We talked about morulas this morning in reproduction. It's a group of cells, okay, that are all just divided out. So here we have strongyles and trichostrongyle eggs. Okay? The next kind of a nematode is a spiroid. The spiroid eggs are somewhat oval or rectangular with a thin, smooth shell, and there's a stage inside of them that almost looks like a larva. Okay? It almost looks like there's a larva in there. And that would be the spiroid eggs. Then, go ahead.
I have a question about the stage. Are you talking about like the stage of life, or actually, like if you know how it goes through like meiosis, mitosis, like division? Is that?
Oh, that's it. That's what you see.
Okay.
The morula stage.
Okay.
Okay. Then we have the trichiroid eggs. They are elliptical, and on each end there's a plug. Okay? Sometimes the shell is really smooth on the outside, and sometimes it's kind of rough looking. The contents on the inside is a single cell. It looks a lot like that one up there, but the shell is different. Okay? So those are typical nematode eggs. You guys need to know which one's which. Okay? Now, life cycle. When we're looking at the life cycle, we need to understand how important it is to understand life cycles. This is how we tell how they're transmitted, how they develop, but it also tells us a lot on how we can treat them and when to treat them. Nematodes can have a direct life cycle. Direct life cycle, like we talked about in the first parasitology lecture, means there's no intermediate host. But nematodes can also have an indirect life cycle with at least one intermediate host, but maybe multiple. Depends on the worm. A simple life cycle for a nematode is pretty straightforward. First, there's an egg, and then that single cell starts to develop into a morula. Then there's a tadpole, and then there's a first stage larva. So basically what we're doing is inside the egg, we've got one cell, then it starts to divide, then it creates this thing that's kind of like a larva, but then it goes to a larva. That larva is the L1 stage. Then the egg hatches. Once the egg hatches, now we have L2, the L2 stage. The L2 stage will molt or change and become an L3. The L3 phase of nematodes is usually the infective stage. That's when it can get into another animal. Then it goes from L4 to L5. L5 is called the immature adult. And then the immature adult can migrate to wherever they live inside the body. Now they're a mature adult. So you basically can see this as just like a bird. Egg, egg is laid, it starts to develop in the egg hatches, gets bigger and bigger and becomes an adult bird. That's basically what we have with the simple life cycle. The definition of a mature adult means that it is reproducing, which then means, because it's nematodes, it has to have males and females. Now, L1s to L3s could be living in the external environment, out in the grass out there, okay? Or that could be inside of an intermediate host, depending on what kind of parasite it is. The L3 then is going to be into, or get into, the definitive host, because again, that's the infected state. Make sense? Go ahead.
So the L3s to L5s can't live in the external environment?
Nope.
Only inside of hosts?
Has to be in the animal.
Okay.
All right, that's nematodes. Now we're going to look at cestodes. When we look at cestodes, there's two basic types of cestodes. Cestodes can either be the cotylodes or the pseudo-tapeworms, okay? And these things at their very end, or their head, if you will, they have something called a bothria. It's not typical. It's kind of like this enlargement that has little slits along the side, okay? Those are pseudocestodes. Eucestodes, or the true cestodes, have a scolex. A scolex means that it has a head. And on that head, there can either be suckers to attach, or a rostellum that has hooks in it, okay, to attach to the inside of the patient.
The rostellum that's like with a butterfly, they have that, they have schools all like that. Is that a rostellum?
This means what's at the front of them, yeah.
Okay.
Like rostral,
Oh, okay.
same base. Now, cestodes are segmented, okay? Caudal to the head, or the scolex area where they're attached, they have a germinal region. And that means that that's the area that starts to produce the segments. So they start from the front next to the neck, and then as they go further and further back, they get more and more mature. Okay? So here we have the sucker, or the scolex area. There's the neck, and then you can start seeing the segments once you get away from the neck. And as they get further and further away, they become more and more mature, if you will. Now, after that neck, we get what's called the strobilia, or the body. This is where the proglottids, or the segments of the tapeworm are. Each of those little segments is called a proglottid. And they are all identical. They all look exactly the same. Cestodes are hermaphroditic. That means the male and the female is in the same organism, which is very interesting. That means they have both reproduction, so they can actually cross-fertilize or self-fertilize themselves. All they have to do is get one proglottid in contact with the other, and now they can be fertilized. Or if there's multiple tapeworms, they can cross-fertilize. Okay? So it doesn't need to have two sexes. They've all only got the one, which is essentially both in one. Make sense?
The process of fertilization is, you guys, is it like completely random, or is there something that sets it off, or is it like time?
So once they get mature,
It's just,
once they move far enough down the tapeworm, then they become sexually mature. You can tell because they have eggs in them. There's the trigger for that fertilization to occur. Okay? And again, as they go from front or by the neck region and further back, they become more and more mature. By the time they get way back at the end, they start falling off. And when they fall off, they get outside the body, and that's how they can be transplanted. Okay? Now, when you look at their general shape or morphology, they are kind of flattened in a dorsal ventral direction, so they look kind of like a ribbon. The size variation is tremendous as well. For example, Echinococcus granulosus, which is a really bad tapeworm, is this guy. That thing around it, that's a paperclip. Okay, so now you can see how small those tapeworms are. And that's a really bad tapeworm to get. And then we get Diphyllobothrium latum. That can be up to 12 meters long. Okay? 12, that's longer than from here to the door. Okay? That's one tapeworm. Tapeworms do not have a GI tract on the inside. They absorb their nutrition through their outside layers. Okay? So they're bathed in nutrients. Those are absorbed right across their skin, if you will. Okay? So they don't need to have a GI tract. That's pretty efficient. So basically, what do? Reproduce. That's pretty much it. Okay? Because that's the only organs they really have to do. The reproductive tract is in every single proboscis. Every one of those has its own reproductive tract. And so each one of them starts to produce eggs. The further and further that they move back, they start to produce those eggs. Okay? Once they are reproducing, then they become gravid. Once they're gravid, the reproductive organs that are in there disintegrate, they just go away. And then all you have left is an egg sac full of tapeworm eggs. And that's it. And that's what usually comes out the south end of the northbound animal. Okay? The eggs, when they are inside, they are in the larval stage. And this is what's interesting about tapeworms. When you look at tapeworm eggs, they have something inside of them that's pretty easy to identify. That's called the hexacanth. And the hexacanth looks like hooks. And you can see it right there. There's the hexacanth for that tapeworm. There it is for this one. This one has a whole bunch of eggs in it. Each one has its own hexacanth. The one that doesn't have one is the pseudophilidium tapeworm. This guy. And that just has this little trap door on the end of it. Okay? So that would be like Diphyllobothrium latum. It's a little bit different. Now, if we look at the different eggs, by looking at them, you can then identify which tapeworm it is. Okay? If we look at the piriform apparatus, that's where it's kind of pear-shaped. Okay? If we look at Dipolidium caninum is the usual tapeworm we're looking at. That's the one that has multiple eggs on the inside, and each one has its own hexacanth. Then we have Taenia. Taenia is the one that has the wide, thick shell that's kind of corrugated, and there's one hexacanth on the inside. Okay, and then again, the pseudophilidium or the Diphyllobothrium latum has the operculated egg, and the hexacanth is in there, but oftentimes you just can't see it. Okay? So, by looking at those eggs, I can tell you what tapeworm it is. And that's what you guys need to be able to do. Okay? Now, when we look at the pseudo tapeworms, they're kind of different when it comes to their life cycle. These things, eggs are passed individually, but they're passed into a water environment. Okay? Once that egg comes out and gets into the water, it hatches. And as it hatches, it becomes chorocydium. Little dinky thing that looks like it's kind of swimming around out there. And it develops a little bit, and then eventually that chorocydium is going to be ingested by something. Generally, what it's going to be eaten by is a little copepod arthropod. Okay? little tiny thing, little crustacean looking thing. And inside of the copepod is going to develop into something called a procercoid. Then along comes a fish and eats the little crustacean. The crustacean is eaten, and inside the fish now, that coricidium, or now the procercoid, develops further into a pleurocercoid. This is the effective stage. Okay? Now, the second intermediate host comes along and it ingests it. Ingests the fish. That's pretty easy. Fish are eaten by a lot of things. Once it gets eaten, now that intermediate thing comes out as a tapeworm, attaches to the intestinal wall, and it starts to develop its strobilia. Okay? So, this is the typical diphyllobothrian type of a tapeworm, or the pseudotapeworm. Notice what the hosts can be. Lots of different animals can be hosts, even us. Guess how we get it? Eating poorly cooked fish. So, anybody want sushi?
I'm kind of now.
Right? It all depends on the kind of fish you're eating. I love it. I used to live in Japan. It was one of my favorite things to eat. A long time ago. But I still eat sushi, but I just make sure I eat the right kind. It doesn't have that possible ingestion of those copepods. Okay? All right, now we get to the true cestodes, the true tapeworms. These things, the proglottids are tasked, not just the individual egg. That egg sac basically comes out the south end, and thousands of those eggs can be released. They can be all over the place. The egg is then going to be ingested by the intermediate host. Okay? So here we have eggs that are released. These things are eaten or ingested by an intermediate host. In this case, the flea larva. The flea larva eats that egg, and inside of that flea larva, it starts to develop. And it develops into a metacestode, which has different stages. A cysticercoid, cystocircus senuris, hydatid cyst, or a tetrapterygium. It all depends on which type of tapeworm, as to which one of those metacestodes there are. Okay? And they're different depending on the tapeworm. Now comes along the second host, usually the definitive host. And it's going to eat the adult flea. Okay? In adult flea that was on that ate the tapeworm egg sac now has the tapeworms in it. They develop into the flea. Dog comes along, cat comes along, or a human comes along and eats the flea. And now the adult can form. Attaches to the inside of the intestine, and now you get tapeworms. Sure. You ever been in a place where there's a lot of fleas? Anybody ever been and seen where fleas are all over the place? You ever seen a little kid that touches a dog and then puts their fingers in the mouth? See, I told you, this is really important stuff to know. Now, obviously, we are incidental, but the dog or the cat, they're the definitive host. What happens when a flea bites a dog? And how do they scratch sometimes? With their teeth. Turn around and start chewing on it. Guess how the flea gets in the mouth? Right? All right, any questions about cestodes? Now we get to trematodes. Trematodes, generally, when you look at them, they're unsegmented and they're flat, kind of like a leaf. Okay? A lot of different size variations. We can have Manaphytes salmonicola, which is that guy. basically that big for a trematode or a fluke. Or we can have fasciole magna. That's this guy. Okay? About this big. Trematodes can either be leaf-like in their shape, kind of flat, or they can be worm-like, especially the schistosomes. These are schistosome flukes. Okay? We'll talk more about them later. There are monogenetic flukes. Monogenetic live their entire life in one host. The host can be a fish, it can be an amphibian, or a reptile. Notice what's not on that list. Mammals. Mammals don't get monogenetic flukes. But we do get diagenetics. Okay? Diagenetic have 2 to 4 obligate hosts. Obligate means it's absolutely necessary for their life cycle. Potentially, we are paratenic hosts. What's a paratenic host again?
Transport.
Pardon?
Transport.
Transport. Just transports it from one point to another point. That's where we can get involved. Flukes are hermaphroditic. That means both sexes are in one. Okay? They have both male and female organs inside that one body. Except for blood flukes. Blood flukes are the schistosomes. They don't have that flat-like appearance. These are the ones that are inside the blood vessels. The fertilization for those that are hermaphroditic is self-fertilization, or it can be cross-fertilization. Obviously, the schistosomes have to go through sexual reproduction because there's males and females. Now, if we look at the internal structures of trematodes, they have a GI tract, which is very unique. At the oral end of it, they have a sucker, but then they also have a pharynx. The sucker helps them to stick there. Then it goes through an esophagus, and then it goes into what's called blind ceca. There's no way for them to eliminate it from another opening, because there isn't another opening. So when they want to eliminate waste, they belch it out, they regurgitate it out. Okay, so it comes back out their mouth or their pharynx. Flukes do not have a body cavity. So basically what you're looking at is reproductive organs and their GI tract. At the head area, they have an acetabulum, and right on that acetabulum is a sucker. And that's what they suck onto the tissues with to hold on. If we look at the diagenetic trematode life cycle, and what's diagenetic again? 2 to 4 obligate hosts. Okay? Eggs are passed. When they get externally, they get into the water. And in the water, the egg hatches and becomes a miracidium. Now, miracidium swims around out in the water. Okay? Now, once that miracidium's out there, the indirect life cycle, and what does indirect mean again? Intermediate hosts. Okay? So, the intermediate host to start with is usually some kind of a mollusk, like a snail. Comes along and eats this guy. Or this guy can penetrate right through into the snail. Okay? Once it gets into that snail, then it forms what's called a sporocyst. And then it goes through the Redia stage. And inside there, now all of a sudden it starts to develop to where it's got a cercarial stage. Then the cercarial options, when they come out of the snail, okay, now they're out in the water again. Now they can either penetrate directly through the skin of the next host, or they can attach to vegetation, insist on vegetation, and just sit there and wait until some animal comes along and eats it, eats the vegetation that has the miracidium on it, or excuse me, cercaria. So cercaria can either penetrate right through the skin, or they can insist on vegetation and then be eaten. Once they're inside the definitive host, then they can mature. If they penetrate to a second intermediate host, if they get into it, they form a mesocarry inside of there. And then the second host is eaten by the definitive host. So again, now we have possibility of here eating the vegetation. Anybody ever eat watercress? Guess what? Gonna have on it. Okay? Or what if you eat the secondary host? If you eat a piece of meat. Okay? Now you can get it. Trematodes are commonly found in the digestive system. Common locations to find trematodes include the rumen or the reticulum, again depending on the kind of trematode. They can be in the intestines, they can be in the liver, even into the bile duct. Those are all flukes up there in this cow liver. They just cut the cow liver and then all of a sudden they're out there, okay? They can also be in your lungs, okay? And then they can get up into your lungs and then they can pass through other directions. And then some flukes, like the schistosomes, are inside the blood vessels. You can actually see them, okay? Eggs of trematodes, when you look at them, they're operculated. Okay, that means they have the little trap door right there. They look a lot like that Diphyllobothrium cesto we talked about. Okay? All right, now we get to acanthocephalans. The acanthocephalans are the thorny-headed worms. This is a very, very small phylum. In domestic animals, there's only 2 species. Most commonly, we see the acanthocephalans in fish or aquatic birds. And we'll talk about that because then they can get into you. General morphology, they look kind of long and round like a nematode. Those aren't really segments, they're cylindrical, but they're kind of like an earthworm. If you've ever looked at an earthworm, there's little parts on the earthworm. It's not a different segment. It's all connected, but there's little divisions in the body. Okay? Size variation anywhere from about that big to about that big. Depends on the species. Now, when you look at their proboscis or their nose area that's retractable, on the end of it, it's usually spine covered. Got lots of little hooks on it. And those spines are directed caudally as far relative to the tape or the worm itself. So that means when that thing goes in, you can't pull it out because the hooks are going back. Okay? It's kind of like the teeth of a boa constrictor or a python. They're directed caudally in the body. Okay? Acanthocephalans, they don't have a GI tract. They're kind of like a tapeworm that way. They just absorb the nutrients across their outer surface. Acanthocephalans are dioecious. The female of the species is bigger than the male, and they are really efficient at reproduction. 250,000 eggs per day they can produce. The life cycle, the eggs, when they are hatched out, they contain something called an acanthor. The acanthor is eaten by some kind of a bug, an arthropod usually. And one of the bugs that commonly eats them is the dung beetle. Okay? Inside the dung beetle or inside the arthropod, the egg hatches, and it hatches and develops into an acanthella. And then a cystacanth with an inverted proboscis. It's not outside yet. Then something comes along and it eats the beetle or the arthropod. Inside that secondary host or second host, it attaches to the intestine and becomes an adult. Okay? Any questions about nematodes? Generally speaking, cestodes, trematodes, or acanthocephalids. Hopefully you're starting to get some pictures here in your mind's eye of how you can get into these life cycles. Okay? And that's kind of an important concept in veterinary medicine.
So, most of the time in intermediate hosts, there's no like clinical signs, right? There's like no effect until it gets to its definitive?
It depends on the parasite.
Okay.
All right, so now we got to talk about the little ones, the protozoans. When we're looking at the protozoans, the first order to look at is the mastigophorans. Mastigophoran protozoas are flagellated. That means they have a little flagella or something on them that allows them to move around. The trophozoite is the modal form. And because they have that flagella, they have to be in a liquid environment. Well, that could be anything from your blood to the lymphatic fluid or even the cerebrospinal fluid. Okay? Pathogenicity of these organisms is variable depending on which type you're looking at. And there's lots of different types of flagellates out there. We have Leishmania up there, trypanosomes, which lives inside your blood. We have trichomonas or tritrichomonas here, histomonas, and probably the most common is Giardia. Okay? The next protozoa to look at are the sarcodinians. The sarcodinians are the amoeba-like organisms. Their trophozoite form is also modal, but how they move around on a solid surface is they put out a pseudopod or an extension. It attaches to a solid surface, and then the rest of it kind of pulls along behind it. Okay? These trophozoites, they will change into a cyst if the environment gets so bad that they can't survive. They just insist, and they stay there and stay there until those conditions change to where now they can come out of that cyst and start their process again. Depending on the type of sarcodinian, then pathogenicity is variable. We don't know for sure unless you know what kind of parasite it is. Some of the parasitic amoebas include Entamoeba histolytica, Acanthamoeba species, or Entamoeba coli, and then Ageria fowleri. Those are all amoeboid type of parasites. The next protozoa is the Coeliophorus. These are ciliates, different from the flagellates. The trophozoite form has the cilia all around it. When you look at it, there's little hair-like extensions all the way around it. And those hair-like extensions kind of move. In order for the trophozoite form to move, it has to be in a liquid environment. They also can form a resistant cyst if the environment gets adverse enough and they can't swim around anymore. The parasitic forms of the Coeliophorus are really pathogenic. Some of the pathogenic ciliates include Balantidium coli or Ichthyopteris multifilus. Okay? Anybody ever have a fish tank? You ever get ick in your fish? Guess what that is? Ichthyopteris multifilus. Okay, they have that parasite. That's why they call it ick, because of its Ichthyopteris. Not because it's disgusting. It's because of the parasite. Okay? Now, the next protozoas are the apicomplexes. And of this kind of protozoa, almost all of them are parasitic, or parasitic to all of the animals out there. They can be intestinal parasites, they can be intracellular parasites inside your blood, or in your reticular activating system, or reticular endothelial system. Their life cycles depends on the host physiology, and that depends on the type. So here we have different types of these parasites, and it all depends on where they're at as to what's going on with them. but it's integrated into the physiology of the host, so it can be very, very effective or efficient. Some of the apicomplexes that are really pathogenic are Hemosporidia. The Hemosporidians are things like Plasmodium vivax, Hemoproteus, and leukocytozoans. Well, everybody knows what these are, right? Plasmodium vivax. Never heard of malaria? Okay, that's pretty bad. Then we have the pyroplasms. The pyroplasms include everything from Babesia to Filaria or Cytoxylin felis. Okay? Notice where you find them. They're in the blood. The first one is the blood up there of either a reptile or an amphibian or a bird. Okay? Because the red blood cells are nucleated. Okay, the apicomplexes again. There's some really pathogenic ones. One of the most commonly pathogenic ones is coccidia. And there's lots of different kinds of coccidia out there. Everything from Mymeria to Cystisospora to Toxoplasmodontii, okay, to Sarcocystis, encephalidozoan, and then hepatozoan. Those are all protozoal parasites that can cause major problems, okay? Especially in humans if the human is immunocompromised. Okay, that's your introduction into endoparasites. Anybody have questions? Anybody afraid yet?
No.
Okay? Go ahead.
So, Cryptosporidium is a type of coccidia.
Yeah.